Welcome to this overview session of cardiac anatomy and physiology. By the end of this session, you will understand the anatomy and physiology of the heart. The session should be complemented by any good anatomy and physiology book. Let's start and consider the location of the heart. It is in the chest, and the heart is off-center, lying to the left at an angle of approximately 45 degrees. This is a cross-sectional diagram of the heart. I'd like you to have a go at labelling the diagram. On a piece of paper, note the labels 1 to 11. You should pause the presentation and restart once you have completed the labelling. Hopefully now you'll have a piece of paper with the labels 1 to 11. Let's go around. We will start with the pulmonary valve. This is the outflow valve of the right ventricle. Deoxygenated blood passes through this and into the pulmonary trunk to the pulmonary circulatory system. Next is the left atrium. This is the collecting chamber for oxygenated blood returning from the lungs. Note the two ostia form the pulmonary veins returning blood to the atrium. The mitral valve is the one-way valve between the left atrium and the left ventricle, preventing backflow from the ventricle to the atrium. The left ventricle is the pumping chamber that sends blood around the systemic circulatory system. This is a high pressure area with a very strong muscle. The intraventricular septum is the dividing muscle between the left and right ventricles. It contains some of the conduction system for the heart. Papillary muscles are attached to the chordae tendineae, and these in turn are attached to the mitral valve and the tricuspid valve. The right ventricle is the pumping chamber for deoxygenated blood to the lungs through the pulmonary valve. The tricuspid valve is the one-way valve to the right atrium. The tricuspid valve has 3 leaflets, while the mitral valve has 2 leaflets. The right atrium is the collecting chamber for blood returned from the systemic system through the vena cava. And finally, we have the aortic valve, the outflow valve to the aorta. Now let's have a look at the anterior surface of the heart. Again, pause the presentation and have a go at recalling these labels. Okay, let's have a look through the labels. The superior vena cava is returning blood from the head, neck and arms to the right atrium. The aorta is the main artery leaving the heart to the systemic circulatory system. The pulmonary artery is taking deoxygenated blood to the lungs. The initial portion of the pulmonary artery is called the pulmonary trunk, and then this splits into the left and right pulmonary artery. The right and the left atrium are the collecting chambers on the right and left side of the heart. The right ventricle is mostly on the anterior surface of the heart, while the left ventricle is mostly lateral and posterior. The heart itself is a muscle and requires its own blood supply. The right coronary artery is seen here, originating from the aorta, and here is the left anterior descending artery, which is a division of the left main stem artery. Now let's have a look at the posterior surface of the heart. Again, pause the presentation and make notes, and then continue when you are ready. The aorta, the pulmonary artery, and the superior vena cava we have already seen. On this view, we can see the inferior vena cava returning blood from the abdomen, pelvis and legs. The posterior of the right atrium and the left atrium are seen here. The pulmonary veins return oxygenated blood from the lungs. We have a superior and inferior pulmonary vein from each lung. These are important when considering atrial fibrillation. A lot of electrophysiology treatment for atrial fibrillation takes place in this area. We can now appreciate the size of the left ventricle in the posterior heart. Here we have a continued view of the right coronary artery, and we can see the circumflex artery. Remember the left anterior descending artery was a division of the left mainstem artery? The other division of the left mainstem artery is the circumflex artery. Finally, we can see the cardiac vein, which is returning venous blood to the right side of the heart via the coronary sinus. The pericardium is the membrane that surrounds and protects the heart. It is composed of two layers separated by a narrow cavity. The inner layer is firmly attached to the heart wall and is known as the visceral layer or epicardium. The outer layer is composed of a relatively inelastic connective tissue and is termed the parietal layer. This fibrous layer prevents distension of the heart, thus preventing excessive stretching of the heart muscle fibers. The cavity between the two layers contains a small volume of fluid which serves as a lubricant, facilitating the movement of the heart by minimizing friction. The wall of the heart consists of three layers: the epicardium, which is the external layer, the myocardium, which is the middle layer, and the endocardium, which is the inner layer. The epicardium is thin, it's a transparent outer layer of the wall and is composed of delicate connective tissue. The myocardium is composed of cardiac muscle tissue, making up the majority of the cardiac wall, and is responsible for its pumping action. The thickness of the myocardium mirrors the load to which each specific region of the heart is subjected. The endocardium is a thin layer of endothelium overlying a thin layer of connective tissue. It provides a smooth lining for the chambers of the heart and covers the valves. The endocardium is continuous with the endothelial lining of the large blood vessels that attach to the heart. Cardiac muscle fibers are shorter in length and larger in diameter than skeletal muscle fibers. They also exhibit branching, which gives an individual fiber a Y-shaped appearance. A typical cardiac muscle fiber is 500 to 100 micrometers long and has a diameter of about 14 micrometers. Normally, there is only one centrally located nucleus, though occasionally a cell may have two nuclei. The sarcoplasm of the cardiac muscle is more abundant than that of skeletal muscle, and the mitochondria are larger and more numerous. Cardiac muscle fibers have actin and myosin filaments arranged in the same way as skeletal muscle fibers and possess a well-developed T-tubular system. In contrast to skeletal muscle, cardiac muscle does not fatigue, cannot be repaired when damaged, and is regulated by the autonomic nervous system. Although cardiac muscle fibers branch and interconnect with each other, they form two separate functional synthia, one for the atria and another for the ventricle. The end of each fiber in a network connects to its neighbours in an irregular inverse thickening of the sclerolemma called intercalculated discs. The discs contain dermosomes, which hold the fibers together, and gap junctions, which allow ions to travel between cells and permit the rapid propagation of action potentials. Consequently, excitement of a single fiber at either network results in stimulation of all other fibers in the network. As a result, each network contracts as a functional unit. So let's have a look through the electrical conduction system of the heart. In what we're going to call sinus rhythm, the impulse will originate at the sinoatrial node. This has an intrinsic rate of 60 to 100 beats per minute. Impulses will then travel across the right atrium and through Bachmann's bundle, which is this branching across the top here, to the left atrium. Once the impulse has travelled across the atrium, it reaches the atrioventricular node, or the AV node. The AV node is a gatekeeper between the atrium and the ventricle. In the normal heart, this is the only communication channel between the atrium and the ventricle. Later on, when we look at some re-entry tachycardias, we will see that there can be abnormal pathways between the atria and the ventricle. From the atrioventricular node, the impulse then comes into the bundle of His and quickly divides into the bundle branches, the right bundle branch and the left bundle branch. We should consider the left bundle branch as the origin of this branch because it quickly divides into a posterior fascicle and an anterior fascicle. Notice the difference between the posterior fascicle and the anterior fascicle. The anterior fascicle is a single strand, whereas the posterior fascicle is more of a fan shape. For this reason, it is much more common to see anterior fascicle blocks as it is to see posterior fascicle blocks. The terminal ends of the conduction system are the Purkinje fibres. Now, as I've just alluded to, there are various different pacemakers within the heart, and the myocardium is able to beat on its own using its own action potential. Hopefully, the impulses originate at the sinoatrial node, and this will give a rate of 60 to 100 beats per minute. Should there be a reason why the sinoatrial node is not commencing electrical activity, then the AV node or the atrioventricular node can take over. However, the rate is a bit slower at 40 to 60 beats per minute. In some very slow rhythms, the impulse is generated in the terminal ends of the conduction system, and rates that are as slow as 20 to 40 beats per minute could be started here. For a final activity, what I'd like you to do on a piece of paper is draw the diagrams we've just discussed. Draw the cross-sectional diagram of the heart and draw the conduction system of the heart and label these. You can look back at the earlier parts of the video to compare your results. One good way to learn and recall anatomy and physiology is to teach it. So find some willing colleagues or family members, maybe bribe them with a bit of cake and undertake some teaching, because this will really help you anchor your knowledge in this area.